Hey everyone. For anybody who doesn't know me, my name is Kyle Cromer. I'm a PI at UCSF in the Department of Surgery, cross-appointment bioengineering and therapeutic sciences. And I think I'm the last talk today, ending with genome editing. So we'll talk about really where this stuff is going in the future in the clinic. So before I talk about genome editing, I have to talk about the genome. It was not all that long ago that we got the double helix structure of DNA that's now famous. It took us 50 years to get the full complete genome. This established the normal human genome. I had folks ask me if this then allowed us to, understand all different types of disorders and, is it done? And I said, well, this established the control that you then compare everything else to. You can sequence folks with rare disorders and understand what typos they have in the DNA. This is called on par with the moon landing. Truly foundational, really not all that long ago, 20 years ago. And then what followed was the next 10 to 15 years was a golden age of driver mutation discovery. This is understanding the typos that cause these common and rare disorders, of which I played a small part, sequencing tumors. And what remains for clinical genetics is finding increasingly rare drivers of increasingly rare diseases or parsing complex genetics. And so I was left, this was my grad school training in Rick Lifton's lab, doing sequencing. And I was left wondering, where do we go from here? Fortunately, the answer was given to me in the form of a talk on CRISPR in 2012. So it took us from really reading the genome as a Microsoft Word document, which I got comfortable doing in grad school, to now truly editing the genome as if it's a Microsoft Word document. Obviously, then it becomes a whole different conversation about delivery, editing efficiency, really making sure that you're getting the intended edit to the intended cell type to correct the disorder. And so what this looks like, just in a more granular detail, is this is a Cas9 protein. This is the most common thing when people talk about CRISPR. This is the original one characterized. It's a nuclease, so it creates double-strand breaks on this double helix. It essentially it can bind this guide RNA that you then can feed to it that allows it to perform this scanning Control F function across the genome. And when it finds a near or perfect match, it can fully bind here and then initiate cleavage. And so, continuing the Microsoft Word document analogy, this Control F function scans, allows a break to happen. But as you might be aware, most diseases are caused by broken genes. And so there aren't a lot of examples where breaking more things can be curative, but there are a couple. And those really are low-hanging fruits that I'll get more into. There's the copy function. And so what you can do then is pair this Control F cut by also providing at the same time to the cells a DNA repair template. So this is a region of DNA that matches right around the region that you're cutting with even changes up to a 1 base pair resolution. And so what then happens is you use the cell's endogenous DNA repair machinery to grab onto that template and use that to repair this double-stranded break, incorporating your edits with extreme precision. And this is why it really does look like when we're drawing up, in the initial planning stages of introducing an edit, like a Microsoft Word document, like I showed on the previous slide. However, what I would say is that most diseases are caused by loss of function mutations, various ones scattered throughout a gene, sometimes on different alleles for the same patient. And so single point mutations that could be curative would be viewed as different drugs, as Tippy mentioned, by the FDA. If you swap out the guide RNA, you have to treat it as a different drug. And so while there are a couple examples of this, such as base editing for the sickle cell disease mutation, you need additional flexibility to be able to change, to compensate for loss of function mutations scattered throughout a gene and kind of a universal one-size-fits-all strategy. And so this is an example of some of the work a schematic of it that I did training with Matt Porteous at Stanford, where the logical next step from sickle cell disease was beta thalassemia, where you have the same gene mutated, but various mutations depending on the patient. And so what we did was use essentially a paste function. You can still do the same Cas9 guide RNA mediated double-stranded break, and then flood the cell with a DNA repair template that actually has an integration wedge between the homology arms to essentially allow the cell to repair that region and then insert whatever sequence you want down to exact precision. And so this is really a way to treat disorders that that are caused by these loss of function mutations scattered throughout. But also, this is a strategy that can only be done right now using this ex vivo workflow. In vivo site-directed integration is something that will be left for the future. But really just to highlight that CRISPR gives us truly unprecedented control over the genetic code, and we're just beginning to scratch the surface on what this can do. To talk about the hemoglobinopathies, which is, was in the news just lately, which I'll talk about. There were curative options preceding genome editing and gene therapy even. This is in the form of a matched donor transplant, where an immunologically matched donor can donate their bone marrow, essentially isolate the stem cells and transplant them. Unfortunately, less than 20% of patients have a suitable matched donor. And because it's not a perfect match, it carries with it the risk of immune rejection and graft versus host disease. To compensate for that, gene therapy and now genome editing allows the patient to be their own donor in the form of autologous bone marrow transplants. This involves removal of the bone marrow isolation of the hematopoietic stem cells, correction of these with editing or gene therapies, and then retransplantation, which does address the shortage of matched donors. And because the patient is their own donor, there's this low risk of immune rejection or graft versus host disease. For this technology that's now been integrated into this workflow, Jennifer Doudna, just across the bay, and Emmanuel Charpentier won the Nobel Prize in 2020 for CRISPR. And it then has been in the clinic even before they won the Nobel Prize. The first patient was dosed with a CRISPR-based therapy. This was at CRISPR Therapeutics, and this was to treat sickle cell disease. It was a way to, I mentioned that most diseases are caused by broken genes, and so there aren't a lot of examples about where you can break more things and correct a disorder, but sickle cell disease is one such disease. It mimics naturally occurring mutations that lead to hereditary persistence of fetal hemoglobin. Sickle cell disease doesn't present until the first year of life when beta globin in the green line turns on, and gamma globin, the fetal precursor to that that's responsible for forming hemoglobin tetramers to carry oxygen throughout the bloodstream, downregulates. This is what naturally happens, but in folks with this hereditary persistence of fetal hemoglobin, they keep their gamma globin on. It never actually does turn off because of some broken machinery, but actually in this case, we can leverage this. What you can do is mimic these naturally occurring mutations to break the repressor of fetal hemoglobin. So a double negative reactivates gamma globin and ends up being curative for sickle cell disease. Now over 70 patients have been dosed safely, again, in this ex vivo autologous patient as their own donor workflow. So that was the clinical trial, and now it just got approved in December of last year, and it was truly a landmark for the field. It does show that the FDA is receptive to CRISPR-based therapies, even though we don't have necessarily perfect information about off-target profiles, but in the context of severe disease, the risk profile was worth it. And so folks in the field, Jennifer Doudna being one, called it an extraordinary moment. David Liu, who's pioneered a next generation of editors called base editors, called it a major milestone, certainly agree. Even Joe Biden chips in and says that it was a significant medical advancement, which it certainly was. So what this does is take your sickled red blood cells, and now that they are producing functional hemoglobin in this fetal precursor gamma, they have healthy, even circular red blood cells. And just shown here, it's a little too small to read, but just durable reactivation of fetal hemoglobin that's persisted up to 2 years in longitudinal data. So this really does seem to be working quite well. So we do have to ask, is sickle cell done? Now really conceivably every patient who suffers from this could be treated. But there is a catch, of course, is that it is highly effective, but the price tag is over $2 million. What I will say is that, the patient cost for their lifetime does exceed this, but a $2 million therapy definitely has its limits in terms of curative capacity, because it's also not even factoring in the additional cost of several months of hospitalization, transfusions, and chemotherapy. That chemotherapy is needed to clear out the bone marrow niche before you re-engraft your edited cells to bump up your engraftment rates to reach curative levels. And so the question then remains, will this not only impact the 100,000 folks in America with sickle cell disease, but more than 90% of cases occur in Africa and India. And so affordability is a big question here. And so while half the field seemed to be praising, CRISPR Vertex for this landmark, the other half was wondering how people would be left to pay for this. And the first person, publicly at least, to come out and talk about her curative experience with CRISPR Therapeutics did say that it is a horrible thing knowing that something is out there that can cure your disease, but you can't access it due to affordability. So I have to ask, what can we do to lower cost? These are ex vivo editing therapies. Of course, cells are never easier to manipulate and edit than when you can pull them out of the body, isolate the stem cell populations, do your fancy editing to them, and retransplant them. So the lowest hanging fruit are sickle cell disease, thalassemia, the severe combined immunodeficiencies. However, what I will say is that most diseases don't have stem cell populations amenable to extraction, editing, retransplantation. Think Huntington's, Duchenne's, cystic fibrosis, and many more examples. And then, just like I mentioned on the previous slide, even for diseases that you can isolate the stem cell populations, edit, and retransplant, it may never be cheap enough to treat the majority of patients suffering from these diseases. Extended hospitalization, also high morbidity myeloablation, that chemotherapy to clear the bone marrow for the edited cells, hands-on development time, and then the fact that the hemoglobinopathies affect the developing world. So Francis Collins, former head of the NIH, quoted, "A genetic cure would have to be given directly to patients rather than by treating cells removed from patients and genetically transformed in the lab. That could avoid the resources needed and complications that can occur with ex vivo therapies." And so the NIH has put its money where its mouth is. The Gates Foundation also believes heavily in this. And so what this would look like, the alternative would be direct injection of your editing tools in vivo delivery vectors. This could even be done outside the context of a hospital if necessary. But I'm not trying to make it sound too easy. This is very difficult, and we are just now starting to do this. I was really surprised when this paper came out personally. I think we were 5 years ahead of where I thought that we were when this paper came out. This is a landmark too. It's in vivo editing by Intellia that was mentioned earlier. This was liver editing. It's another example of a disease you can fix by breaking more things. This is in a disease called TTR. And what it looks like is Cas9 mRNA and guide RNA. So these are your editing tools packaged in a lipid nanoparticle. It's fairly generic, and so where does it go but the liver? It's very easy relative to other cell populations to transduce and edit the liver. And so what happens is this lipid nanoparticle gets taken up into hepatocytes. The mRNA then codes for your Cas9 protein that then combines with the guide RNA, homes to the nucleus, scans the genome, and then finds this TTR gene and cuts it. And so what this involves is then direct delivery to cells without toxic conditioning. You don't have to pull cells out of their native environment to then edit them. Well, I'll say the liver is the easiest target, but many groups are working to target other tissues, including Tippy's group as well. This is, in this form, it's hit and run editing. This is not persistence of your editing tool. So ideally it can go in, do the edit that you want, not the edit that you don't, any edits that you don't want, and then it will degrade and initiate a permanent change as long as you're hitting stem cell populations. One particular feature is LNPs, and there's been some nice data on this, that they can possibly be redosed compared to viral vectors. You could potentially redose them until you hit therapeutic levels. And then the final thought on LNPs packaging Cas9 is an LNP plus mRNA. In terms of manufacturing, there are major concerns that viral manufacturing, not only they cannot be redosed, but also it's very, very expensive. You have to essentially replicate a human infection in cells, isolate that virus, purify it, quality control. Whereas LNPs and mRNA can be manufactured at great scale, as was done successfully during the COVID pandemic. And these vaccines, while the formulation is, of course, going to be quite different, at a chemical level, they're quite similar and was delivered to the world at $3 per dose. So I do think this is eventually how you could make it cheap enough. I'm not saying they'll be that cheap, but there is a path to making them affordable. And so where does editing meet precision fetal medicine? This is in the form, this is essentially the slide that Tippy showed before, but this is in utero genome editing, things that you would diagnose prenatally, things that manifest prenatally, or might manifest postnatally, but just have particularly accessible stem cell populations where you can make a bigger impact at a lower dose than treating them postnatally could also be justified. And so for our part, along with a larger group, UCSF, led by Tippy, including then myself, Dana Laird, Deepak Srivastava, at Berkeley, Jennifer Doudna, one of the discoverers of CRISPR, Fyodor Urnov, Petros Niran, Murthy, and UC Davis and Emory. We have, I mean, it's going to take a village to do this, but I think we have a great team assembled. And then finally, I also don't want to end with such negativity about Casgevy, because it was a huge landmark. It did show the FDA will approve CRISPR-based therapies. Of course, the very first one would have been ex vivo. This was always going to precede in vivo editing, and in vivo is really difficult. There are other challenges. Now, when you isolate your HSC population in a dish, edit them, they're easy to edit, and you're not putting other cells in the body at risk of editing that won't impact your disease. And so ex vivo, while expensive, does solve that problem. In vivo editing runs the risk of being delivered to all the other cell types of the body that could have unintended effects. Also, Casgevy defined what a curative genetic change could be for sickle cell disease. And so what you can do is take these editing tools and then package them in the most effective delivery vehicles and deliver them in vivo. So we've already defined the payload. Now we just need the delivery vehicles. So, and then I think I want to end on the statement that affordability and accessibility still needs to be at the core of considerations when advancing this technology. For too long, gene therapy and genome editing has been bashed for being just $1 million therapy, and it's going to take some major technological innovations to change that.